In this video, we are going to be discussing weak interactions. And by the time you're done watching this video, what I want you to be able to do are the following. I want you to be able to describe the difference between weak interactions and covalent bonds or molecular bonds. I want you to be able to describe the importance of weak interactions in biology. I want you to be able to understand and identify examples of ionic interactions, hydrogen bonds, and hydrophobic interactions. So let's start with the difference between weak interactions and covalent bonds or molecular bonds. And first, let's take a look at some weak interactions. Here we have a weak interaction. This is called a hydrogen bond. Here's another weak interaction. This is a hydrophobic interaction. And here's another one here. This is an ionic interaction. And notice one interesting thing that we'll learn a lot more about a little bit later is these interactions are all occurring within a single molecule. This molecule happens to be a protein, and a protein is a long chain of amino acids. So these weak interactions don't necessarily have to occur between two different molecules. They often occur within the same molecule as this example here. Now, weak interactions are definitively different than covalent bonds or molecular bonds. So for example, here, this bond between this carbon and this carbon, this is a covalent bond. The bond between this sulfur and this sulfur, that's a covalent bond, okay? Weak interactions are about 10 to 40 times weaker than covalent bonds, okay? Which means that within an aqueous solution, at physiological temperatures and physiological pHs, weak interactions are constantly being broke and being reformed, okay? They're made, they're broken, they're made, they're broken. And it's this weakness that is the important nature of these interactions. Whereas covalent bonds, under sort of physiological conditions, unless you have an enzyme catalyzing the reaction, these covalent bonds are simply not going to just spontaneously break the way these weak interactions do. Well, what is so important about weak interactions? Well, what's important is they are weak, that in physiological conditions, you can sort of break them fairly easy, or they're broken fairly easily. But that doesn't mean that there isn't some sort of strength there, and the strength comes in numbers. So here is a molecule of DNA, and a molecule of DNA is made up of two strands, and these two strands are connected to one another not by covalent bonds, but by weak interactions here, weak interactions here, weak interactions here. So even though each individual weak interaction is weak, when you add up millions and millions and millions and millions of them between a molecule, they can be quite strong enough to keep the two strands of DNA from spontaneously separating from one another. But because each individual weak interaction is weak, there are regions of DNA that can separate. Okay, not the whole giant molecule, but just regions. And this ability to separate strands of DNA in different regions is important for activities like transcription and DNA replication. Okay, so weak interactions are absolutely essential for biology. Let's take a look at some other examples. We talked about this. So the importance of weak interaction in biology is important in the base pairing, DNA, the two strands, proper folding of polypeptides, of the folding of a protein into its proper structure is dependent upon many weak interactions. The specificity of binding of an enzyme to its substrate are dependent on these. So for example, here we have an enzyme. This is chymotrypsin. This is the active site, the pocket that binds to the substrate, and here's the substrate. And we've got an important interaction going on here between the enzyme and the substrate. In this case, it's a hydrophobic interaction that we'll describe in just a few moments. Here's another example. This is trypsin. Trypsin is an enzyme, and this all in purple is trypsin. Here is the active site. That's going to be the place that binds to the substrate, and here's the substrate. Now, one of the reasons that that very specific substrate binds here is because of weak interactions. In this case, ionic interactions between one of the amino acids of trypsin and an amino group here of the substrate. Okay. We have weak interactions being important for fueling the catalytic power of enzymes, of the binding of antibodies to their antigens, and the binding of ligands to various cellular receptors. Each one of these we are going to be learning about in more detail a little bit later on in the course. So weak interactions are absolutely essential for biology. We're going to learn about 3 weak interactions that will come up over and over and over again. We have a hydrogen bond shown here, which involves a polar hydrogen and some other polar atom within a molecule. We have the interaction, ionic interactions, which involves two ions, oppositely charged ions that stick together. And then we have hydrophobic interactions that depend upon 2 hydrophobic molecules being excluded from the aqueous solution. Let's take a look at each one of these in more detail. First, ionic interactions. Very straightforward. Ionic interactions require oppositely charged ions. Remember, an ion isn't a partial charge. An ion has either more electrons than protons or less electrons than protons. So you're either going to have a net negative charge or a net positive charge. Okay, so here we have an anion, a negatively charged ion, and a cation, a positively charged ion. And within an aqueous solution, the interaction between these opposite charges is there, but it's weak. It's not like an ionic bond, right? When you have water in the situation here, it's going to weaken the interaction between these 2 oppositely charged ions, but they are still strong enough to be absolutely essential for many aspects of biology. Next, hydrogen bonds. Hydrogen bonds require, as the name suggests, a polar hydrogen. That means a hydrogen that isn't an ion, right? Okay, but it has a partially positive charge, and then some other polar atom. So let's take a look. Here we have our hydrogen bond, and notice there is that polar hydrogen. How do we know that it's polar? Because it is covalently bound to an atom that is much more electronegative than it. Right? So this oxygen is more electronegative than this hydrogen, and therefore the electrons that are being shared between these 2 atoms fly more commonly, more oftenly around this oxygen and less frequently around this hydrogen, which means this oxygen is partially negatively charged and this hydrogen is partially positively charged. Beautiful. There's our polar hydrogen. That's 1/2 of the equation. The other half is a partially negatively charged other thing. Here, our ammonia is a nitrogen connected to 3 hydrogens. And we know this nitrogen is going to be partially negatively charged because we know nitrogen is more electronegative than the hydrogens that it's covalently bound to. That means the electrons that are shared between these atoms are more frequently flying around the nitrogen and less frequently flying around the hydrogens. So this nitrogen becomes partially negative. It's not an ion. It's not a full negative charge. It's a partial negative charge. So this partially positively charged hydrogen sticks to this partially negatively charged nitrogen, forming a hydrogen bond. Okay, and lastly, one of the hardest maybe to understand because it's sort of a misnomer. Hydrophobic interactions aren't really a direct interaction of sticking together of two molecules. Let me try to explain that a little bit better. What hydrophobic interactions require is non-polar molecules. Those are molecules that are made up of atoms, all of equal electronegativities, such that you don't get the sort of polarity that we've seen in these polar molecules. We don't get any partial negative or partial positive charges. So let's say these two molecules are that. They are a non-polar molecule. And you need them to have hydrophobic interactions, you need your water. Okay? And here are the water molecules. Now we know that water molecules are polar, and so as you'll learn in another video, the water molecules form tons of hydrogen bonds with other water molecules. They stick to each other quite a bit. And so we've got all of these hydrogen bonds, hydrogen bonds, hydrogen bonds forming between water molecules, but because the nonpolar molecule is nonpolar, because it can't interact with water, these molecules get excluded from the water-water interactions. And because they're excluded from those interactions, they sort of lump together. Now, there's nothing inherent of sticking these two things together. The reason they're kind of clumped together is because they're being pushed out by all of the water molecules interacting with one another. And so this exclusion of non-polar molecules from the interactions of water with each other or water molecules with other polar molecules, that exclusion is what we refer to here as hydrophobic interactions. Okay, so those were the three types of weak interactions I want you to focus on. And now I have a practice problem for you. I have three molecules and I have proposed some hydrogen bonds. A hydrogen bond between that hydrogen and that oxygen, one between this carbon and this hydrogen, and one between this nitrogen and that hydrogen there. Okay. R just means the rest of the molecule, so you could ignore R, really. And my question to you is, which one of these proposed hydrogen bonds is possible? Why don't you go ahead and pause this for a moment and try to solve it. Okay, so we're back. Well, let's take a look at this. Let's answer this question. Remember, every hydrogen bond needs a partially positive hydrogen and a partially negative something else. So let's take a look at each one of these. Is this oxygen partially negative? Well, to figure that out, we need to see what it's connected to. It's covalently bound to a hydrogen. We know oxygen is more electronegative than hydrogen. Therefore, that oxygen is partially negatively charged. Well, how about this hydrogen? In order for that to be a proper hydrogen bond, that hydrogen has to be partially positively charged. Let's see if it is. It's covalently bound to this carbon. Carbon and hydrogen have equal electronegativities. Therefore, the electrons are flying around equally, so this hydrogen does not have a partial positive charge. This is not a possible hydrogen bond. Let's take a look at here. Here we have, this hydrogen. Is it partially positively charged? It's connected to an oxygen. Oxygen is much more electronegative. Yes, that hydrogen is partially positively charged. Fantastic. Now we need a partially negatively charged atom. Well, here's carbon. Is it partially negatively charged? Well, it's connected to hydrogens. Carbons and hydrogens have equal electronegativity. Therefore, that carbon does not have a partially negatively charged. And no, this is not a possible hydrogen bond. All right. That leaves us with the third and final possibility. Let's go through each one of those. Do we have a partially positively charged hydrogen? There's our hydrogen. It's covalently bound to an oxygen. Oxygen is much more electronegative than hydrogen. Therefore, that hydrogen is indeed partially positively charged. Let's take a look at nitrogen. Is it partially negatively charged? Well, it is covalently bound to hydrogens. We know nitrogen is more electronegative than hydrogen. That means the electrons spend more time around the nitrogen. That means it is indeed partially negatively charged. Partially negatively charged, partially positively charged, they connect to one another via a hydrogen bond. So the answer to this question is C. What you should gather from this are anytime you have carbons and hydrogens connected together, they don't have polarity. No positive, negative, no partially positive charges, no partially negatively charges. And molecules that consist primarily of just carbons and hydrogens are called hydrocarbons, and they are non-polar molecules, and we'll look at those in more detail in a later video.